We are going to learn about biotechnology principle and processes. In today's video, I will break down few tips about biotechnology that can make NEET or UPSC preparation more effective, whether you are just starting or deep in revision. So make sure to watch till the end and don't forget to subscribe if you like this video. Manipulation of genes by man is a fast emerging technology which started with the formation of recombinant DNA molecules. So this branch of science has been named as recombinant DNA technology or genetic engineering or biotechnology. So what is the biotechnology? In biotechnology, there is a manipulation of genes by whom? By man. It is a fast emerging technology which is started with the formation of recombinant DNA molecules. The origin of biotechnology is as old as human civilization. Example, fermentation is the oldest biotechnological technique discovered by people by prolonged soaking of grains before cooking. The next example is it could have been production of vinegar, curd, etc. So biotechnology is a technological employment of microorganism, plant cell, animal cell to generate products and services which are useful to human beings. Thus, it include all those processes in which natural strains of microorganisms and cell lines are being used in obtaining products like cheese, wine, vinegar, bread, etc. So these processes are now, however, excluded from biotechnology or rather referred to as old biotechnology. These processes are referred to as a old biotechnology or excluded from biotechnology. So based on this old biotechnology, powerful industries have been set in the recent years to produce a variety of products in fairly large amounts, such as food items, bread, idlis, etc., dairy products, yogurt, cheese, butter, alcoholic beverages, beer, whiskey, wine, non-alcoholic beverages, tea leaves, coffee beans, organic acid like lactic acid, enzymes, vitamins, hormones, amino acid, vaccines, steroids, etc. These are included under the old biotechnology. Now move to that modern biotechnology. Modern biotechnology is the use of genetically modified organism or genetically engineered plant and animal cell in developing production technologies that not only enhance their productivity but also give rise to new products. During the last 2 decades, there have been great advancement in recombinant DNA technology, polymerase chain reaction, cell culture, fusion, gene cloning, DNA fingerprinting, etc. So, due to these different kinds of development, the term biotechnology is currently used in a restricted sense to refer to only those processes which are concerned with genetic manipulation to achieve the same on a large scale. So, the last we have to get the product on a large scale. So, a new definition of biotechnology has been given by EFB, both which is combining both traditional and modern biotechnology, which is combining both old and modern biotechnology. Definition given by EFB, European Federation of Biotechnology. Biotechnology is an integration of natural science and organism cells part thereof and molecular analogs for products and services. So some of the biotechnological products and processes are recombinant DNA vaccines, which we will learn in the application of recombinant DNA, which we will learn further. Synthesis of a gene and introduction of it into a target cell or organism, gene therapy, in vitro fertilization for production of test tube babies, production of many biological compounds. So there are different types of biotechnology. So the types are given here: red, green, white, yellow, blue, gray, gold biotechnology. Red biotechnology is a health branch and responsible according to Biotechnology Innovation Organization BIO. With the help of red biotechnology, there is a development of more than 250 vaccine and medications such as antibiotics, regenerative therapies, and production of artificial organs. So red biotechnology is associated with 250 vaccine medication, and the most important thing there is, with the help of red biotechnology, there is a production of artificial organs. Green biotechnology, It is used by the farmers, as the name suggests, green. So it is used by the farmers, more than 13 million farmers worldwide, to fight pests and nourish crops and strengthen them against microorganisms, extreme weather events such as drought and frost. White biotechnology, the industrial branch works to improve manufacturing processes, the development of biofuels and other technologies to make industry more efficient and sustainable. Yellow biotechnology is focused on food production. For example, it carries out research to reduce the levels of saturated fats in cooking oils. Blue biotechnology, this exploits marine resources to obtain aquaculture, cosmetic and healthcare products. In addition, it is a branch most widely used to obtain biofuels from certain microalgae. Gray biotechnology purpose is the conservation and restoration of contaminated natural ecosystem. So gray biotechnology is also very important, which is helpful in the conservation and restoration of natural ecosystem, contaminated natural ecosystem through, as mentioned above, bioremediation process. So with the help of bioremediation process, the technology uses gray biotechnology. Gold biotechnology is also known as bioinformatics, where the technology is associated. It is responsible for obtaining, storing, analyzing, and separating biological information, especially that related to DNA and amino acid sequence. So biotechnology deals with the techniques of using live organism. Live organism forms or enzymes from organism to produce products and processes useful to human. So these are the products which are useful to human. Making curd, bread, wine, which are all microbe-mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organism to achieve the same on a large scale, and this is included under the modern biotechnology. Modern biotechnology using genetically modified organism was made possible only when man learned to alter the chemistry of DNA. So man learned to alter the chemistry of DNA, and with the help of modern technology, there is a recombinant DNA process will take place. So with the help of this genetically modified organism, it's helping to alter the chemistry of DNA and construct recombinant DNA. This key process is called recombinant DNA technology. The process is known as a recombinant DNA technology, and the or genetic engineering. This process involves the use of restriction endonuclease, different types of enzymes. The enzymes names are restriction endonuclease, DNA ligase, which is also known as a molecular glue, appropriate plasmid or viral vectors to isolate and ferry the foreign DNA into host organism, expression of the foreign gene, purification of the gene product, that is the functional protein. At last, we have to get the functional protein in this process, and finally making it suitable formulation for marketing. Downstream processing. Large scale production involve use of bioreactors. So these are the processes we will learn in the next video. That topic is process of biotechnology, about the genetically modified organism whose DNA has been altered through genetical engineering. Example includes pigs, chickens, cow, and fish that have been modified for increased milk or meat production, disease resistance, or to produce a specific protein. So other example includes zebra fish also and transgenic goats also. Biotechnology is technology that utilizes biological system, living organism are part of this to develop or create different products. Brewing and making bread are example of this old biotechnology. So these are all the traditional processes which utilize the living organism in their natural form. So while the more modern form of biotechnology will generally involve a more advanced modification of the biological system or organization. So with the development of genetic engineering in the year 1970s, research in biotechnology developed rapidly because of the new possibility to make changes in the organism genetic material. So we are doing the changes in the genetic composition, that is the DNA of the organism. So biotechnology deals with the industrial scale production of biopharmaceuticals and biological using genetically modified microbes, fungi, plants, and animals. So the application of biotechnology includes therapeutic, diagnostic, genetically modified crops for agriculture, processed food, bioremedies, and waste treatment and energy production. Principle of biotechnology. Principle of biotechnology, there are two core techniques that help in the birth of modern biotechnology. First one is the genetic engineering, or second one is the bioprocess engineering. It is also known as biochemical engineering technique. Genetic engineering, it is the manipulation of genes by man. It refers to artificial synthesis, isolation, modification, combination, addition, and reprieve of genetic material to alter the phenotype of the host organism to suit human needs. So, with the help of genetic engineering, the chemistry of genetic material to introduce into host organism and thus change the phenotype of physical appearance of an organism. Bioprocess engineering, it maintenance of a sterile microbial contamination-free ambience in chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccine, enzymes, etc. So this process is also known as biochemical engineering technique, which helps in the growth of desired microbes or eukaryotic cell in large quantities in sterile medium. The medium is, and the process is tissue culture technique for the manufacture and multiplication of biotechnological products like antibiotics, vaccine, enzymes, medicines, hormones, etc. Conceptual development of principle of genetic engineering. All living organisms reproduce sexually or asexually. Sexual reproduction has more advantages as it leads to more genetic combination and variation. These variation are beneficial to the organism as well as population and can be used in plant and animal breeding programs. However, in case of asexual reproduction, there is a preservation of genetic material and inheritance of exactly same copy. There is no variation. So at times, undesirable characters are expressed during reproduction. Recombinant DNA use of gene cloning and gene transfer isolate and introduce only one or set of desirable genes without introducing undesirable genes into the target organism. So principle of genetic engineering are DNA. For DNA to express when introduced into any other organism, it has to incorporate itself into organism genetic material and replicate. So origin of replication is to start replication, to start duplication, there is one mm, uh, uh, mm, place is needed in the that is known as the origin of replication, a specific sequence of DNA responsible for initiating replication, from which place there is a replication start, and that is known as a ori. Then DNA with origin of replication replicates in new host, and there with the help of vectors, the other allele piece of DNA is joined with the help of enzyme ligase, and it is cut by the another enzyme. So these all process we will learn when we will start the learning of tools of recombinant DNA technology and process of recombinant DNA technology. So DNA with origin of replication replicates a new host and clone making multiple identical copies of any template DNA. So these are the principle of genetic engineering. So however, tradition hybridization procedure often includes inclusion and multiplication of some undesirable genes with desirable ones. To avoid this, genetic engineering techniques have been developed, which involves creation of recombinant DNA, gene cloning, and gene transfer. Thus, technique of genetic engineering include creation of recombinant DNA, use of gene cloning, gene transfer. So, techniques of genetic engineering include creation of recombinant DNA, use of gene cloning, gene transfer. History of recombinant DNA technology. While the structure of DNA was first determined in the year 1953, uh, it was not until the early 1970 years that was the first recombinant DNA molecules were proposed by the means of restriction enzymes. Paul Berg succeeded in proving the possibility to splice and to recombined genetic material in the year 1971. Consequently, Stanley Cohen and Herbert Boyer were able to cut and fuse specifically as located DNA strands. A subsequently submitted patent for recombinant antibody technology was approved in the year 1980. This has paved the way for further development, which are increasingly gaining attention in research and clinical application. So, cloning vector. Cloning vector is also known as here given about the plasmid. So what is a plasmid? Plasmids were discovered by Hayes and Liederwerk in bacterial cell. They are small, double-stranded, closed, circular DNA molecule that occur naturally in the bacteria outside the bacterial chromosome. So in the bacteria, there is a bacterial chromosome also, or the plasmid also. So plasmid is a double-stranded, closed, circular DNA molecules that occur naturally in the bacteria, and they are called extra chromosomal DNA. Plasmids are inherited from parents' bacterial cell to daughter cell and have the capability of self-replication and have the capability of self-duplication. So example of cloning plasmid is PBR322. This is the first artificial cloning vector constructed by Bolivar and Rodriguez. So here the example is given about the cloning vector, then cut DNA fragment cut by the enzyme, then again ligated with the another enzyme, recombinant plasmids. Origin of replication is given here, there ORI, which is also known as the ORI, which is a sequence of bases on which DNA replication starts, where ORI is a sequence of bases on which DNA replication starts. Any piece of DNA linked to this sequence can be made to replicate within the host cell. Under natural condition, each plasmid replicates to produce 20 to 30 copies per cell. So this sequence is also responsible for controlling the copy number of the linked DNA. So it's given about then, and then it is introduced into the plasmid into E. coli host cell divides, and E. coli containing cloned plasmids. So recombinant host cell. Genetic engineering. Genetic engineering involves the techniques to alter the chemistry of genetic material, which we have learned already. This change the phenotype of host organism. And this is the reason asexual reproduction preserves the genetic information, while sexual reproduction permits variation because two parents are involved. Asexual reproduction, only one parent is involved. Traditional hybridization procedure used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes and along with the desired genes. The techniques of genetic engineering, which includes creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation allows you to isolate and introduce only one or set of desirable genes without introducing undesirable genes into the target organism. So these are the three basic steps in genetically modify an organism is identification of DNA with desirable genes, identification of DNA with desirable genes, then introduction of identified DNA into the host with the help of plasmid vector DNA. Here is the cutting, sticky ends is given. What is sticky ends? We will learn. And this is given the blue and white methods. This method is known as a. So the three basic steps in genetically modified organism is identification of DNA with desirable genes, then introduction of identified DNA into the host, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. So the construction of first recombinant DNA molecule was constructed by Stanley Cohen and Herbert Boyer in 1972. They isolated an antibiotic resistant gene from the plasmid of bacterium. The name of the bacterium is Salmonella typhimurium. This gene was responsible for conferring antibiotic resistance. So the Stanley and Cohen and Herbert Boyer in the year 1972 isolated an antibiotic resistant gene from the plasmid, plasmid of the bacterium, and the name of the bacteria is Salmonella typhimurium. The cutting of DNA The cutting of DNA was done. The cutting of DNA was having integral fragment of the antibiotic resistant DNA. So the cutting of DNA was done with the help of restriction enzyme. So DNA cutting was done with the help of restriction enzymes, and the restriction enzyme, which is popularly known as molecular seizure. This piece of DNA, this piece of DNA cut from the plasmid this piece of DNA cut from the plasmid was then linked with another plasmid. So then it linked with another plasmids acting as vector. So it acts as a vector. So this acts as a vector, cloning vector. First, what happened? The cutting of DNA with the help of restriction enzymes. This piece of DNA cut from the bacteria was then linked with the another plasmid DNA, which is known as a cloning vector or vehicle. This linking was done with the help of enzyme, and the name of enzyme is DNA ligase, which joins the end of cut end of DNA molecules. The newly formed DNA then introduced into the this recombinant DNA introduced into the host, and it becomes now the newly formed DNA having integrated fragment of DNA, and it is known as a recombinant DNA. Once the newly formed recombinant DNA is transferred into the bacterium E. coli, it replicates in the host cell. It replicates in the host cell. It replicates in the host cell using the enzyme DNA polymerase. So, enzymes DNA polymerase help in the replication. This result help in the formation of several copies of recombinant DNA having capability of antibiotic resistance. This process of multiplication of gene is called gene cloning. So this method of multiplication of gene is called gene cloning. So the step involved in creating a transgenic organism can be summarized as what identification of DNA with desired genes. The steps are given here. An introduction of identified DNA into a target or host cell, maintenance of introduced DNA in the host, and transfer of DNA to its progeny. Recombinant DNA technology describe a process of genetic engineering that uses enzymes and various laboratory techniques to isolate and manipulate genetic material in various steps. This is possible because DNA molecules from all organisms share the same chemical structure and differ only in the nucleotide sequence. There are different ways to carry out manipulation in an organism genome, all with the aim to improve certain characteristics. So recombinant DNA, it's an important point to note then. Recombinant DNA differs from genetic recombination. Recombinant DNA differs from genetic recombination in that it results from artificial method in the test tube. While the latter, genetic recombination, is a normal biological process that results in the remixing of existing DNA sequence in essentially all organisms. So no need to be confused with there should be no ambiguity that there is a difference between recombinant DNA which differs from the genetic recombination. Recombinant DNA technology describe a process of genetic engineering that uses enzymes and various laboratory techniques to isolate and manipulate genetic materials, and there are having different steps. So DNA molecules from all organisms share the same chemical structure, differ only in the nucleotide sequence. Methods in recombinant DNA technology. There are three methods of producing recombinant DNA technology. First one is transformation, non-bacterial transformation, and phase. Transformation and non-bacterial transformation are similar processes, with the only difference being the use of bacteria such as E. coli for the host. So bacteria such as E. coli for the host for the case of transformation and non-bacterial transformation. While the transformation process uses Escherichia coli to act as a biological framework, the non-bacterial method does not use any bacteria. So non-bacterial method does not use, as the name suggests, non-bacterial. So it does not use any bacteria. Initially, one needs to choose which DNA segment to insert into the fragment, into the vector. The second step is to cut the piece of DNA with the restriction enzyme, restriction endonuclease, with the help of restriction endonuclease, then ligate, and then DNA insert into the vector with DNA ligase. So it's given about this plasmid cut with restriction enzyme, gene of interest, then it ligated recombinant plasmids. The vector is inserted into a host cell which has to be specifically prepared to take up the foreign DNA. Now, about the phase introduction is a process of transfection, which is equivalent to transformation, but this process, a phase is used instead of bacteria. The in vitro process uses lambda or M13 phases to produce phase plex containing recombinates by the means of different selection methods. The generated recombinant DNA can be differentiated from non-recombinant DNA. Okay, so this is about the difference between the phase, about the transformation, non-bacterial transformation and phase. Recombinant DNA molecules are DNA molecules which is formed in the laboratories and such as the molecular cloning to bring together genetic material from multiple sources, creating sequences that would not otherwise be found in the genome in the complete genetic makeup. It is possible because DNA molecules from all the organisms share the same chemical structure. They differ only in the nucleotide sequence within the identical overall structures. So in most cases, organisms containing recombinant DNA have apparently normal phenotypes, normal physical structure. That is, their appearance, behavior, metabolism are usually unchanged. Now, some frequently asked question about recombinant DNA. What is recombinant DNA technology? It is a laboratory technique. It involves manipulating DNA fragments from different organisms to create new genetic recombination that would not be found in nature. It has various applications, including the production of therapeutic proteins and genetically modified organisms. Second question, what are the steps in recombinant DNA technology? So the steps in recombinant DNA technology include isolating DNA from the donor and host organism, cutting the DNA using restriction enzyme, joining the fragments with the DNA ligase, introducing the recombinant DNA into the host organism, and selecting and screening transformed cells. Transformed cell. What about the safety? If we are conducting the and doing the process of recombinant DNA technology in laboratories, so what about the safety? When considering the pros and cons of recombinant DNA, safety is frequently discussed aspect. It is a very important aspect. Generally speaking, the molecules are recombinant DNA molecules and recombinant proteins are not regarded as dangerous. It is not dangerous. However, concern remain about some organisms that express recombinant DNA, particularly when they leave the laboratory and are introduced into the environment or food chain. Such potential safety issues include antibiotic resistance and adverse immune reaction outside the health sector. Concern include the potential of gene pollution of the environment, but also health effects of food from genetically modified organism. However, to ensure the greatest degree of safety possible, all recombinant DNA work needs to be compliant with standards and guidelines set out by the FDA and other regulatory institutions. Apart from safety issues, there is no ethical issue of genetically modified organism, human genome editing, as well as around genetic information in general. What is needed in recombinant DNA technologies? The next question is the DNA sequences used in the construction of what is needed. What are required in the recombinant DNA technology? So the DNA sequence used in the construction of recombinant DNA molecules can taken from where? Can originate from any eukaryotic species, be it human, bacterial, fungal, or mammalian. In addition, DNA sequence that do not occur naturally may be created by chemical synthesis. So we can create it also chemically. The different molecular biology tools used in recombinant DNA technology include DNA isolation and analysis, molecular cloning, quantification of gene expression, determination of gene copy number, transformation of the appropriate host for replicating starting at a selectable marker, or transfer into crop plants and analysis of transgenic plants. Respectively, some of the most important instruments needed are enzymes, gene cloning vectors, polymerase chain reaction, and host organism. Eligible host organism for recombinant antibody expression include bacterial in yeast cell, but also insect or mammalian cell, and mammalian cell are HEK293 cell or CHO cell. So selectable marker is the word is coming here. In addition to ORI, the cloning vector also require the presence of selectable marker, which help in discriminating the transformant from non-transformant, recombinant from non-recombinant. Is recombinant DNA the same as genetically modified organism? No. Recombinant DNA is not same as genetically modified organism. Recombinant DNA refers to DNA molecules formed through combining genetic material from different sources. No need to confuse that. Recombinant DNA means DNA molecules are taken from different genetic materials from different sources, while genetically modified organism or organism whose these are organisms genetically modified, so whose genetic materials has been altered, whose genetic composition has been changed using recombinant DNA technology. What are transgenic organism? These are genetically modified organism. Transgenic organism or organism whose genetic material has been modified by the introduction of genes from another species using recombinant technology. So the method is using is a recombinant DNA technology, and transgenic organism or genetically modified organism whose genetic material has been altered using recombinant DNA technology. These inserted genes can come from plants, animals, bacteria, or other sources. The purpose of creating transgenic organism is often to confer specific desirable traits such as the increased resistance to pest, improved nutritional content, or enhanced growth rates. Transgenic organisms are commonly referred to as genetically modified organisms and have application in agriculture, medicine, and research. Now, next question, question 7. What processes are involved in recombinant DNA technology? Processes are isolating genetic material, desired genetic material, cutting DNA fragment at a specific location, joining DNA fragment by ligation and polymer tailing, inserting DNA into the host cell, selecting and screening the transformed cells. Application of recombinant DNA technology, it is used in medicine, research, identify, map, sequence genes, and to determine their function. It is used to produce human insulin, human growth hormone, recombinant blood clotting factor, hepatitis B vaccine, insect-resistant crops. So this is a table which is given about the human insulin humulins, which is the used for diabetes and discovered by the company that Eli Lilly. Human growth hormone, trade name is Humatrop. Application is pituitary dwarfism, and company developed was Eli Lilly and Genentech. Erythropoietins, the trade name is Ipogen, and application is for anemia patient, and company developed was Amgen. Factor 8, factor 8, which is medicine for hemophilia, and company was Baxter. DNA is 1, which is the trade name is Dornasa alpha. It is for cystic fibrosis, and company was Genentech. Interferons, intron A for hepatitis B, C, multiple sclerosis, and company is sclerosing plug. Tissue plasminogen activator, trade name is Activase. It is for acute myocardial infarction, and company is Genentech. So cloning, now the term is also the coming, there no need to confuse with the recombinant biotechnology along with the cloning. Cloning for the asexual reproduction where we get the exact copy. There is no variation. So cloning is the production of an exact copy of a cell, any other living part, or a complete organism. Cloning of animal was successfully performed for the first time by Ian Wilmot and his colleague at Rosen Institute in Edinburgh, Scotland. They cloned successfully a sheep named Dolly. Dolly was born in the year 1996 and the first mammal to be cloned. So Dolly was the first mammal to be cloned. During the process of cloning, Dolly, a cell was collected from the mammary gland of female Fin Dorset sheep. The name is the female sheep is Fin Dorset sheep, and we have he has taken mammary gland of a female. Simultaneously, an egg was obtained from a Scottish blackface phoebe. The nucleus was removed from the egg. Then the nucleus of mammary gland cell from the Fin Dorset sheep was inserted into the egg of the Scottish blackface phoebe, whose nucleus have been removed. The egg thus produced was implanted in the Scottish blackface phoebe. Development of this egg followed normally, and finally Dolly was born. Though Dolly was given birth by Scottish blackface Eve, it was found to be absolutely identical to Finn Dorset sheep, from which the nucleus was taken. Since the nucleus from the egg of the Scottish blackface Eve was removed, Dolly did not show any character of the Scottish blackface Eve. So cloning is somewhat transferred into an alien piece of alien organism, would not be able to multiply itself in the progeny cell of the organism. But when it's get integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA, alien means new piece of DNA, has become a part of chromosome, which has the ability to replicate, which has the ability to duplicate. In chromosome, there is a specific DNA sequence called the ORI, origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be a part of the chromosome, which has a specific sequence known as a ORI. Thus, alien DNA is linked with alien DNA is linked with the origin of ORI, so that this alien piece of DNA can replicate, can duplicate and multiply itself in the host organism. Thus, cloning or making multiple identical copies of any template DNA. So, this is about the animal cloning. So, Dolly was healthy clone of the Finn. This is the image, Finn Dorset sheep, and then Scottish blackface sheep. Since Dolly's several attempts have been made to produce cloned mammals, however, many die before birth or die soon after birth. The cloned animals are many a time found to be born with several abnormalities. Biotechnology in India, the remarkable march of India into the world of bioscience and technological advances began in the year 1986, Mr. Rajiv Gandhi period, during Rajiv Gandhi periods. In the year 1986, a separate department for biotechnology with the Ministry of Science and Technology, vision attaining new height in biotechnology research, shaping biotechnology into a premier precision tool of the future for creation of wealth and ensuring social justice, especially for the welfare of the poor. Biotechnology has made a huge impact on Indian agriculture, healthcare, industry, and environment on one hand, with raising global standing of India. So the National Biotechnology Development Strategy aims to make US dollar 100 billion bioeconomy by the year 2025. Biotechnology in India, there is a many sector. It is expected to play an important role in shaping the country's rapidly developing economy. So bioeconomy is driven by research, innovation in biotechnology and life sciences, and enabled by technological advances in engineering as well as in information science. India's bioeconomy is on its way to achieve the USD 150 billion target by the year 2025 and over 300 billion by 2030. So India is one of the top 12 biotechnology destination in the world and the third largest in Asia Pacific. Biotechnology is the technology of tomorrow and can leverage the wealth of India's bioresources, its unique biodiversity, its 7,500-kilometer long coastline and vast agricultural residues. So it is a trending career option among youth due to increased attention to tools like genome editing, synthetic technology, microbial bioresources, enzymes, and metabolic engineering. Startups, because of the growing enabling ecosystem, the number of biotech startups in the country has increased from 50 to over 6,756 in the last 10 years. The government aims to increase the number of biotech startups to 10,000 by the year 2025. Subsector of biotechnology in India, according to the revised India Bioeconomy Report 2023, India's bioeconomy sector stand at $137 billion, contributing 4% to the national GDP and employing over 2 million people. So institutional facilities for biotechnology in India in the year 1988, National Biotechnology Board was constituted by the government to strengthen the indigenous capability, and in this newly emerging discipline in the year 1986, Department of Biotechnology also support research and development innovation. The National Center for Microbial Research established to conserve India's vast microbial diversity and explore its biotechnological potential for industrial and health application. Indian Biological Data Center, it is India's first national repository for life science data and Autonomous Institute of DBT. So these are the institutional facilities for biotechnology in India. The government initiatives of biotechnology in India, there are different initiatives that the government has taken to improve the biotechnology in India. So some of the initiative taken by the government to further promote the sector are as follows: National Biopharma Mission, to accelerate biopharmaceutical development in the country, Innovate in India I3 program. As part of this mission to create an enabling ecosystem to foster entrepreneurship and indigenous manufacturing in the sector. Biotech Kisan. So it launched a Biotech Kisan Innovative Science Application Network in 2017. Its goal is to bring science laboratories and farmers together to develop and apply innovative solution and technology at the farm level. Atal Jai Anusandhan Biotech Missions. So these are the maternal and child health, antimicrobial resistant vaccine for infectious disease, food in nutrition and clean technologies. One Health Consortium in the year 2021 to survey important bacterial, viral, parasitic infection of zoonotic and transboundary pathogen. Biotechnological Industry Research Assistant Council established by DBT and is aimed at strengthening and empowering emerging biotechnology enterprises to undertake strategic research and innovation. Biotech parks also offer facilities to scientists and technology demonstration, pilot plant studies to accelerate the commercial development of biotechnology, startup landscape. So the biotech sector in India registered more than 1,000 new startups in a single calendar year in FY22. So funding has helped, BRAC funding has helped more than 1,000 startups. So there are challenges also associated with biotechnology. Despite the boom in the biotech industry, there are some grave challenges faced by this domain: ownership and equitable access, ethical concern, field of uncertainty, security threats, impacts on environment. So security threat, how a technology that may be used in laboratory to artificially produce organism have raised concern about the possibility of biological weapons development, impact on environment. It can create a potential negative environmental and ecosystem function result of agricultural biotechnological technology, development of pest and virus resistant crops, and impact on wild species as well as pollinators. Ethical concern: biotechnologically can produce unanticipated consequences that cause harm or dehumanize people. For example, complementary DNA is a genetically designed entity that can be patented.
Obtaining such DNA and altered DNA sequence that can be sold for profit raises numerous ethical concerns about human life. It has ownership and equitable access also, so which protects ownership rights in new biotechnologies. It can also prevent many people from benefiting, particularly in developing countries. So these are the challenges associated with biotechnology. So these are the summary. Biotechnology given humans several useful products by using microbes, plants, animals, and their metabolic machinery. It has made it possible to engineer microbes, plants, and animals such that have novel capabilities. We have created genetically modified organism with the help of biotechnology, genetically modified plants, genetically modified crop plants, then immense impact in the area of healthcare. Since the recombinant therapeutics are identical to human proteins, they do not induce unwanted immunological responses. We have produced human insulin is made in bacteria at its structure is absolutely identical to that of natural molecules with the help of biotechnology. Transgenic animals are also used to understand how genes contribute to the development of disease by serving as model for human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's disease. Gene therapy technology is also developed with the help of to treat with many hereditary disease with the help of biotechnology, with the help of gene amplification. Viruses that attack their host and introduce their genetic material into the host cell as part of the replication cycle are used as vector to transfer healthy genes or more recently portion of genes. So in the next video, we will learn application of biotechnology in various fields. Next video, application of biotechnology in medicine, and then in agriculture, in animal husbandry, in food processing, in environment. So we will learn so many topics related with the biotechnology.